Hey everybody, welcome back to General Biochemistry. We are in the second part of the course. We're introducing the rest of the macromolecules, and we're also going to learn some concepts that are related to, looking at metabolism, so some generalities. So right now we're looking at chapter 7, which is Carbohydrates and Glycobiology. We're only covering the first two sections. We're not going to get too deep into glycobiology, but I do want you to be able to recognize some of the big players in terms of carbohydrates. And we'll talk a little bit about the purposes of carbohydrates, how they're stored. These are the key principles that you should be able to expand upon after you watch this lecture. So always make sure to go back to this slide and read these and make sure that you understand them. We'll start with monosaccharides and disaccharides, giving you some terminology when we're talking about carbohydrates. So in general, a carbohydrate is an aldehyde or ketone that has at least two hydroxyl groups. Or it can be something that would yield this compound on hydrolysis. And we'll talk about that at the end. That's more like the big storage molecules. Lots of carbohydrates have the same empirical formula, CH2O, and it's just some multiple. So that's what the N is. You multiply by 1 or 2 or 6, whatever it is, and that tells you how many carbons, hydrogens, and oxygens are in that carbohydrate. There's different classes of carbohydrates. Monosaccharides, we're talking about simple sugars like glucose, and they have one polyhydroxyaldehyde or ketone unit. So you're going to need to remember those functional groups here to recognize an aldehyde or ketone. And if you're good with your prefixes, mono should tell you 1. Okay. Then we have oligosaccharides, short chains of monosaccharide units, and they're joined by glycosidic bonds. You'll also see or hear glycosidic linkages. They're the same thing. Within the oligosaccharide class, you can have disaccharides, and these are just oligosaccharides that have two monosaccharide units. A good example that most people are familiar with is sucrose, or table sugar, and that is when you take D-glucose and D-fructose and you put a link between them. There's a glycosidic linkage between them. Then we've got polysaccharides, which are the big sugar polymers. So we're talking 10 or more monosaccharide units. These are the storage polysaccharides. So cellulose, glycogen, amylose. Those are all polysaccharides. So think sugar storage when it comes to polysaccharides. We talked about stereochemistry a little bit. We're not worried about it too much when it comes to the amino acids, but stereochemistry is absolutely key when we're talking about carbohydrates because all the enzymes that deal with sugars are stereospecific. So sugar stereoisomers are they're present simply because you have carbon atoms where hydroxyl groups are attached, and we've got all these chiral centers. So remember, a chiral center means that you've got a carbon that's got 4 different things bound to it. So that is an example of a chiral carbon or a chiral center. And you see lots of those in sugars. Remember that you can have all the, something pointing out at you, something pointing towards the back, away from you, to the left, to the right. So all that stereochemistry matters when it comes to enzymes because they are stereospecific. This is really important when we get to looking at glycolysis. So just keep that in mind. Within monosaccharides, we've got two families, aldoses and ketoses. You can probably guess that aldoses have an aldehyde, and ketoses have a ketone. When we're looking at the backbone of a monosaccharide, and this is just the linear form here, we've got unbranched carbon chains with a single bond linking all the carbon atoms. So that is our unbranched carbon chain with all single bonds. One of those carbon atoms has a double bond to oxygen. And that is what defines whether or not it's going to be an aldose or a ketose. In our example here, we have the carbonyl carbon at one of the ends. So there's a hydrogen also attached to that carbon. Whenever you see both of these, this is going to be an aldose because it has an aldehyde. If you see maybe one of these carbons or one of these carbons and it has a double bond to oxygen, that would be a ketone. That would be a ketose. All the other carbon atoms are bonded to a hydroxyl group, and they also have hydrogen, but we don't really care about that. One of the defining features is all the carbons have some kind of oxygen bound to them. So just to go through the aldose and ketose identification again, the aldose, the carbonyl carbon, is at the end of the carbon chain, so an aldehyde group. And if you have an internal ketone, it is a ketose. You will need to know the difference, and you'll need to be able to look at sugars and determine if it's an aldose or a ketose. So these are just some examples, but you'll need to be able to do that with sugars that you've seen and sugars that you've never seen before too. So make sure that you know the difference. Trioses are the simplest monosaccharides. They've only got 3 carbons in their backbone. Now, usually we talk about how sugars have all these chiral centers. Glyceraldehyde is an exception. It only has 1 chiral center. Dihydroxyacetone is a ketose. And you can see how the naming works here. Aldo for aldose, and then triose because it's got 3 carbons. We're not going to focus too heavy on names, but I do want you to be able to look at a name and understand it. But you won't be responsible for naming things per se. These 2 trioses you're actually going to see again when we get to glycolysis. So you'll see a lot of repeat offenders. That's why I kind of call this section like meet the macromolecules, because you're going to see a lot of structures that you're going to see again in metabolism. Then we've got tetroses and pentoses. If you're good with your prefixes, you'll know tetra or tetro could be 4, and pent is 5. And there's some examples here. We've got ribose and deoxyderibose, and they each have 5 carbons, so they're pentoses. When you look at the top, you see the aldehydes here, so they're aldopentoses. Then there's hexoses and heptoses. Again, prefixes. If you remember them from chemistry, then you'll be good to go. Hex is 6. Hept is 7. We're showing you a lot of different sugars. A little bit of an aside, what makes sugar sweet? Well, on your tongue, you have all of these receptors. And the 2 genes that encode for the receptors that tell your brain, hey, this is sweet, are TAS1, R2, so TAS1 receptor 2, TAS1 receptor 3. When the sugar hits your tongue, it has to bind to the receptors and cause them to dimerize. Dimerize just means that you have, 2 proteins that are coming together. So at the top of this figure, we've got some kind of a sugar, glucose, sucrose, fructose. You can also have all those sugar substitutes. So sucralose, which is Splenda, aspartame, all these other things. They bind and cause these 2 different receptors, the R2 and R3, to dimerize. There are interactions between the receptor and the sugar that brings them closer together so that they touch. And when they touch, on the inside, they relay to a GTP binding protein, hey, we've got something out here. Something has bound. And that sends a signal cascade that ultimately results in your brain receiving that, or generating that sweet electrical signal. All of this is dependent upon stereoisomers. So there's D and L, just like with amino acids, same thing with sugars. You have to have a steric match. So if you don't have the right stereochemistry, you're not going to bind that receptor. So when you think about all the sugar substitutes that are made, they have to be sterically compatible with these receptors. Otherwise, you're not going to taste sweet. You might be activating some other taste that you don't really want to. You don't want your coffee, well, some people might want their coffee spicy or salty. I, however, I haven't had coffee in years. But when I was an avid coffee drinker, I didn't want hot as in spicy coffee. I didn't want salty coffee. If I wanted it to be sweet, just a little hint of sweetness. So that's what those taste receptors do. They bind to a molecule and relay a signal into the cell that results in your brain generating a signal that tells you sweet. Okay. Monosaccharides have asymmetric centers. The only exception is dihydroxyacetone. So it does not have an asymmetric center. But all monosaccharides contain at least 1 chiral carbon atom. We talked about enantiomers very, very briefly in chapter 3 when we introduced the amino acids. But this is just a reminder that in an enantiomer, we're talking about 2 different isomers that are mirror images. So if you put a mirror in front of a molecule, the way that mirror is represented with all of the stereochemistry, all the functional groups going in and out of the plane and left, right, that is an enantiomer. So if you have a lot of chiral centers, you're going to have a lot of stereoisomers. I don't need you to know how many stereoisomers you can get based on how many chiral centers there are. We're not concerned about that. Just understand that you can have a lot of different stereoisomers simply by having more than one chiral center. Whenever you're talking about carbohydrates, we have to talk about Fischer projections. Probably covered this in organic. I'm not going to worry about you generating Fischer projections, but I do want you to recognize certain molecules in their Fischer projection. But it's the 3D representation of a sugar structure on paper. There's a certain way that you draw it to represent bonds that are going in and out of the plane of the paper. So the bonds that are drawn horizontally are indicating that bonds are projecting out of the plane of the paper, or if you're looking at a screen, like you're watching this, out of the plane of your screen. And then there's the vertically drawn bonds, and they project behind the plane of the paper or the screen. Again, you won't be drawing Fischer projections, but you're going to see a lot of them. This gives you another idea of what an enantiomer is. So we're looking at glyceraldehyde, and it is checking itself out in the mirror. You can see how just because you're looking at a mirror image, you can see the difference in which groups are pointing out, which groups are pointing back, that kind of deal. You're not going to be responsible for identifying are these enantiomers or not. This is not an organic chemistry class, but you need to be familiar with these concepts because these are terms that you're going to see, and sometimes the reasoning for why something, an enzyme does or doesn't work is based on the stereochemistry. So you need to be well versed in the vocabulary. When we're dealing with sugars, we got a lot of carbons. And you have to know how the carbons are numbered because when you read papers or when you look at enzyme mechanisms, you're going to be talking about chemistry happening to particular carbons. The carbons are numbered at the beginning, at the end, near the carbonyl group. So where that aldehyde is, or the carbon that's closest to the ketone, that's where we're looking to start numbering. These are all the aldoses that you'll probably run into. I'll highlight the ones that I want you to recognize. So I would like for you to recognize glyceraldehyde, ribose, glucose, and galactose. Those are the 4 aldoses that I want you to recognize. You don't need to be able to draw the Fischer projection, but you'll need to recognize these molecules in their Fischer projection. In terms of the ketoses, I want you to recognize fructose. So only the ones that are highlighted are the ones that you're responsible for recognizing. So I can give you a sheet of paper and say, hey, label these, and you can tell me D-fructose, and we're only worried about the D, not worried about L configurations. They're not biologically relevant. So we're looking at Fischer projections and we're looking at these linear molecules, but sugars can actually close up and form a ring. In an aqueous solution, aldotetrosis and all monosaccharides with at least 5 backbone carbon atoms, they actually are in their cyclic structure. So there's covalent bonds that form between the carbonyl group and one oxygen of a hydroxyl group. The term that we use to describe these molecules are hemiacetals or hemiketals, and then acetal or ketal. The hemiacetal, the hemiketal, they're derived when you have a reaction between one of those alcohols and the aldehyde or the ketone. So it's when that happens for the first time. So you're taking a linear carbohydrate and you have that reaction with the alcohol and the aldehyde or the ketone. So it's the carbonyl carbon where it happens. And I'm going to show you the chemistry in just a second. You form a 5 or 6 member ring if the alcohol and the carbonyl group are on the same molecule. If they're not on the same molecule, then you are making an oligosaccharide. The acetal or the ketal, that happens if you do that same reaction again. So you have another alcohol that attacks a carbonyl group. That is how you form a glycosidic bond. So you have a ring structure that's been created by the first reaction. And then that cyclic structure can then go out if it's a reducing sugar, and it can attack a different sugar that has a carbonyl that is ripe for picking. So what does that look like chemically? I want you to be able to draw this. So pay attention to this mechanism. I've got it for the aldehydes and for the ketones. This is slightly abbreviated, but I want you to understand how the electrons are pushed here. So we've got our aldehyde and an alcohol. Notice that we've just got R2 here because this could happen between two different sugars, so different monosaccharides, or it could be a linear carbohydrate that is doing this reaction to become a cyclized structure. The hydroxyl group, that oxygen has a lone pair on it. And these make a partial charge on the oxygen. It's actually got more electrons than that, but we only need one pair for this. This carbonyl carbon is actually partially positive. And the reason for that is this electron withdrawing group, the oxygen. The electrons are hanging out more with that oxygen than they are with the carbon. So that makes it partially positive. This oxygen is going to attack the carbonyl carbon. And a couple of those electrons are going to go, we're gonna stay with the oxygen. So you're gonna make that into a single bond. And that's how you form the hemiacetal. So the other step that happens after these electrons say, we're going to hang out with the oxygen, is that then those electrons go out and grab a proton. Because that transition state would be pretty unstable. Now this is a reversible reaction. So the hemiacetal can once again kind of break down, and you would just have some electrons do like this, and then like this, and you would kick out whatever that R group is along with the oxygen. If you were to do this same process again, so you have the hemiacetal and then you add another alcohol, it would be the same process. You're attacking the same carbon because it is still slightly positive. It's got that partial positive charge. It's got 2 oxygens on it this time. So there's 2 electron withdrawing groups that make that carbon slightly positive. Same deal for the ketone. So I'm not going to go through and show the arrow pushing. But if I give you two simple monosaccharides, you should be able to draw how you get from the linear aldehyde or ketone to the hemiacetal. And if I give you another one, you should be able to draw getting from the hemiacetal or the hemiketal to the acetal or the ketal. So when we do these reactions and we're cyclizing these sugars, or we're creating glycosidic bonds between two monosaccharides, we are creating stereochemistry. We're creating more chiral centers. And this stereochemistry, the alpha and beta configurations, I do want you to know. So anomers are the isomeric forms of monosaccharides that only differ about that hemiacetal or the hemiketal carbon atom. And I'll show you what that is. The anomeric carbon, which you'll hear that referred to, is the carbonyl carbon atom. So if we're looking at a molecule, this is the anomeric carbon. Specifically, we're going to look at D-glucose and how it can go between its two cyclic forms. Start with the Fischer projection. It's difficult for me to say this morning. And we're going to have the hydroxyl on carbon 5 go up and attack the carbonyl carbon in the aldehyde. As practice, try drawing that reaction, pushing the arrows and coming up with that cyclized form. So you're going to have that attack, and you have two options for stereochemistry. First option, you end up with the hydrogen in the up position and the hydroxyl in the down position. That's alpha. If the hydroxyl group is up and the hydrogen is down, that's beta. So however it helps you to memorize that, you need to recognize these two forms of glucose. When the hydroxyl is down on carbon 1, when it's in its ring form, that is the alpha. When the hydroxyl group is pointing up, that's the beta. The interconversion of alpha and beta anomers is called mutarotation. We've got purinoses and furanoses. We're talking about more terminology for the ring compounds this time. So purinose is 6-membered ring. So glucopyranose, that is glucose in its ring form. Furanoses have 5 carbons in their ring. Purinoses have 6. Haworth projections, or perspective formulas, another representation of a carbohydrate. These are more accurate for representing the cyclic structure than a Fischer projection. So you'll notice that you've got a difference in thickness of these different bonds that are drawn. The bonds that are closest to the reader, so you, me, whoever's looking at this, they're drawn thicker. So the closest bonds are the thicker ones. The ones that are further away are thinner. So that helps in visualizing the molecule itself, and it's more accurate than the Fisher projection. Whenever you see a molecule represented like this, a cyclic sugar, always look for whether or not it's alpha or beta. So the hydroxyl group is in the down position. We're looking at alpha. This corresponds to any ring structure, not just glucose. I also want you to recognize glucose in the Haworth perspective. So in the ring form, I want you to recognize alpha and beta. Glucose is a very important molecule, so I want you to recognize it in all its forms, all of its glory. So we actually have a lot of hexose derivatives. There's the glucose family, and there's all these different derivatives from glucose. I'm not requiring you to know all of those. I do, however, want you to know glucose, and I want you to recognize glucose 6-phosphate. Glucose 6-phosphate is very important. We'll talk a little bit about why that phosphate on there is important towards the end. Or now. Thought that was coming a little bit later. So phosphorylated derivatives of sugars are very important because they are stable at a neutral pH and they have a negative charge. Stable at a neutral pH, that makes sense because our bodies operate around neutral. So you want your fuel molecule to be stable at that pH. But it also bears a negative charge. And that helps to trap the sugar inside the cell. Otherwise, glucose would be free to move about the cabin and just diffuse in and out of the cell. If you're a cell that needs to generate some energy, you want to hang on to your glucose. So as soon as that glucose comes in, you have a kinase that's going to phosphorylate the glucose and trap it so that it can't leave. You can't force it to go through glycolysis, but you can at least keep it around so that your odds are better. There's no membrane transporters to take the phosphorylated sugar from the cell and pump it out into the extracellular space or into the blood. This concept is going to come back to us when we talk about glycolysis, and it's one of the key hallmarks of metabolic pathways, that you have some kind of way to trap a molecule and keep it where you want it so you can do the chemistry. So sugars that are or can form aldehydes are reducing sugars. I used that term a little bit earlier without defining it. Now we're defining it. So reducing sugar will react, right? So there's a redox reaction where the free aldehyde can actually react with copper 2+ under alkaline conditions. This is actually a test that you can do to help narrow down what type of sugar you have. So if you've ever had a class where I've taught a biochemistry class where I taught a lab as well. And part of one of the labs is identifying, there's this whole made up, hey, everybody ate here at this cafeteria on campus, and now these people are sick. This is what they ate. And you're, some kind of forensic person that's analyzing the stomach contents to try to identify, the major macromolecules, protein, carbohydrates, this and that. And so finding the difference between simple sugars and more complex sugars, we're talking about doing things like this using copper and alkaline conditions, looking at whether or not something's a reducing sugar or not, looking at whether or not you've got starches or simple monosaccharides, that kind of thing. So these tests are useful when you're trying to identify things. When you reduce copper 2 plus to copper 1, it forms a brick red precipitate. So if you see that as a result, then you know you have a reducing sugar. Ketosis can tautomerize to form aldehydes, so they're also reducing sugars. You're not responsible. I'm not going to show the mechanism for that. That's something that you should have covered in organic. If that sounds a little bit foreign to you, then you may want to go back to your organic notes and kind of look that over. Again, not because you're going to be responsible for drawing it, but simply because that is something that should be familiar to you. So we've talked a lot about monosaccharides. We've talked about the Fisher projection, the Hayworth projection, or perspective. But now we need to start putting these monosaccharides together to form disaccharides and larger oligosaccharides. That's where the glycosidic bond comes in. It's a linkage between two different sugars, and it's covalent. So we're joining two monosaccharides covalently. When you have the hydroxyl of one sugar molecule react with the anomeric carbon of another molecule, then you're going to form a glycosidic bond. So remember that this sugar that we're looking at, glucose, can open up. So if it opens up, this is carbon 1 right here. And when it opens up, it's going to have that aldehyde. And then it's also going to have the rest of the molecule, right? So all of the carbons and the hydroxyl groups, I just didn't want to draw all that out. So that's what the R is. When you have the closed structure, you still have that hemiacetal. So remember that it has so this is the hemiacetal. And this would be for a cyclic structure. So this is just one carbon. This is carbon 1 on the glucose molecule. So this carbon has a partial positive charge. A lone pair on a different alcohol on a different sugar can come in and attack that carbon. When that happens, you're going to condense and you're going to release a water. Once that happens, you form an acetal, and this is the linkage. You'll notice that there's some numbers here, 1 arrow 4. That means it's 1 to 4. We have linked carbon 1 with carbon 4. So carbon 1 on this molecule and carbon 4 on the other molecule. When you bring an alpha and a beta glucose together, and they're joined by an alpha 1 beta 4 linkage, you make maltose. So I'm not going to require you to know that sugar, but that is something that if you are into bread making or fermentation, maltose is something that you'll run into. So when you have the formation of a glycosidic bond, that sugar is no longer a reducing sugar. So here and here can't do any more chemistry here. We've already got a linkage. However, we've still got this free anomeric carbon. That is called the reducing end. So sometimes when you do a reaction and you bring together two monosaccharides, you will still have a reducing sugar at the end because there's another part of the molecule that can do some chemistry. So if you have a free anomeric carbon, meaning that carbon that is connected to the oxygen here, so that oxygen within the ring, when you go to that first carbon, that's going to be the anomeric carbon. If it's not involved in any kind of a linkage, then it is a reducing sugar. So that's the reducing end of this disaccharide. As you go through your textbook, you're going to see a lot of different symbols and abbreviations for monosaccharides and their derivatives. You don't need to know these, but this is just for your information when you're looking at even proteins or enzymes that are decorated with sugars, particularly ones that are in the membrane or they're extracellular proteins, that kind of deal. They're going to be decorated with all types of sugars. And instead of drawing out all the structures, which is really, space consuming, you've got all of these little symbols that you can use or abbreviations that you can use to indicate the sugars that you mean. So these are common conventions. Again, you don't need to memorize these, but if you're looking at a paper or something that's talking about the structure of a protein and you see these symbols, then you'll be able to recognize them. So there's 3 common disaccharides. Of these, the ones that I want you to recognize are lactose and sucrose. Those are 2 important ones. So lactose is a reducing disaccharide, but sucrose and trehalose are not. You're not responsible for knowing trehalose. But when you look at sucrose, you should be able to convince yourself that it's not a reducing sugar, it is a non-reducing sugar. And with lactose, you should be able to look at it and see that it is a reducing sugar. So be able to identify the reducing end here. I'm going to leave that for you to do so that if you don't understand, you can ask in class. Now we're moving on to the big boys, okay? Polysaccharides. Most carbohydrates in nature occur as polysaccharides. We're talking huge molecular weights. We also call them glycans. So same thing. You can have homopolysaccharides or heteropolysaccharides. The difference between them is that homopolysaccharides only have one monomeric sugar. So think glucose. All glucose. If you have a long string of glucose, then that is a homopolysaccharide. Heteropolysaccharide, we're talking about at least 2 different kinds of monomers. Typically speaking, the homopolysaccharides are going to be the storage forms that you're going to see in biology. They can also serve as structural elements. Heteropolysaccharides are usually some kind of extracellular support. You'll also notice that you can have branching or no branching at all. And if you have something that's branched, then it's a little bit more space saving. And we'll look at some examples of these. Polysaccharides don't actually have defined lengths or molecular weights. Unlike proteins, polysaccharides, when they assemble, they just assemble based on their own kind of intrinsic properties. So when you're making that big starch molecule as a plant, there is no template that says, okay, starch can only be this many, monosaccharides all strung together. So that doesn't exist. Like I said, homopolysaccharides, think storage fuel. So starch in plants, glycogen in animals. We're not going to really talk about plants like that at all, but we will be talking about glycogen in the upcoming chapters. These molecules are heavily hydrated, which means that they are coated in water because they have all those hydroxyl groups. So you have to be able to appease those hydroxyl groups and not just have them poking out into some solution that doesn't have any water. Hydrogen bonding stabilizes these molecules so that they can stay soluble. Starch and glycogen are both polysaccharides of glucose. Starch, however, contains two types of glucose polymers, amylose and amylopectin. And you will need to know these and be able to compare and contrast them. So starch versus glycogen, and know the difference between amylose and amylopectin. So amylose is just long unbranched chains of glucose. They're connected by alpha-1,4 linkages. Amylopectin is bigger. So it takes those amylose strings, and in addition to the alpha-1,4 linkages, you've also got alpha-1,6 linkages, and it's super highly branched. So that's the difference between amylose and amylopectin. Those alpha-1,6 linkages and the highly branched nature of the molecule. Then we have glycogen. It's got alpha-1,4 linkages, and it's also got alpha-1,6 linked branches. So it is very extensively branched, and it's actually more compact than starch. Here's some examples of looking at these starch and glycogen molecules. So for A, what we're looking at is just a lot of glucose molecules that have the alpha-1,4 linkages. Once you finally get to the end of a strand, and let's say that this just had a hydroxyl group here, that would be the reducing end. So think about this in terms of being able to add on to this starch molecule. You have to have a place to do some chemistry to keep adding on these monosaccharides. So the reducing end is where the action happens. In A, what we're really looking at is an example that could be amylose, or we could be looking at a part of, glycogen that is unbranched. But B is a better representation of amylopectin, or glycogen, because we see the 1,4 linkages, and we also see branch points, and that's the alpha-1,6. So that kind of introduces a turn so that you can fold the glucose molecules back in on itself, and that chain can kind of start going in the opposite direction. In C, we're looking at a combination of amylopectin and amylose. So in blue, you've got you can call it an amylose strand, okay, a chain That's just the alpha 1,4 linkages. But if you take those and you kind of roll them up, and you have these branch points with the alpha 1,6 linkages, then you form the greater structure of amylopectin. Those two things together comprise starch. Why would we want to store glucose as a polymer? Well, there's a couple of reasons. One, space. I mean, think about it. You've got all these molecules free to roam the castle. You need to wrangle them in together and store them in one place. Keep them compact and out of the way. But the other one is you don't want to have high osmolarity. Think about if you've ever made Kool-Aid or lemonade or any kind of sweet beverage, when you put too much sugar, which some people might argue, there's no such thing as too much sugar in Kool-Aid. I disagree. But when you're mixing sugar with water, you can add so much sugar that it becomes syrupy. It's gloopy, gloppy, viscous. I don't think that we want our cells to be that way. So when we store our glucose, storing it as glycogen or as a starch helps to reduce the osmolarity. Okay, so you would actually have 0.4 molar glucose in the cytosol, and that would make it so that water would absolutely be trying to enter the cell, and it would cause it to rupture. So instead of having all the glucose as just free molecules, storing it as glycogen reduces the osmolarity so that you don't have an issue of water seeping in and the cell lysing. Some homopolysaccharides actually serve a structural role. So one example of that is cellulose. So if you like trees and plants and stuff, cellulose is probably something you've heard of before. It's very tough, very fibrous, and it is not soluble in water. It's huge in terms of molecular weight. We're talking about 10 to 15,000 linear unbranched glucose molecules. And you don't need to know all of the information about these different examples for cellulose. So cellulose, I want you to know what it is, but you don't need to know how many glucose units it has. I'm not even going to ask you to know the linkage for it. I just want you to know the general characteristics, that it's tough, it's fibrous, it's insoluble in water, and we actually don't have enzymes to hydrolyze the linkage that they have. So we can't digest cellulose. Chitin is another example. So this is a homopolysaccharide that is comprised of acetylglucosamine. And we didn't talk about all of those different derivatives of glucose. But chitin, so if you're a bug person, that's in the exoskeleton. And it's super hydrophobic because it's acylated. And it's water resistant. So for a bug, that's kind of a good thing. You don't want water just, ending up on your insides. You need to be protected from that water, because water is a molecule that can do some damage. We can see it used as a nucleophile, right? So you don't want a free nucleophile running about the cabin. So there are some factors that influence homopolysaccharide folding, but again, there is no template for it. These are just some guidelines for making sure that it folds in a similar pattern, right? So the 3D structure is stabilized by weak interactions. Where have we heard that before? So you've either got interactions with water and you make a ton of hydrogen bonds, or you have interactions between molecules. So hydrogen bonding is super important here. Just like with proteins, hydrogen bonding helps with solubility and it helps with keeping the molecules stable. There's rotation about both of the CO bonds. So remember that there's an oxygen and then there's carbon on either side when we're looking at the cyclic structure. And so rotation about these is actually pretty limited. And they're limited because of all the other constituent groups that are around them. So you can't really get free rotation of those bonds. Another example of these long chains of polysaccharides is peptidoglycan. So bacterial cell walls are very tough to penetrate. That's one of the reasons why having bacteria that are resistant to antibiotics is such a big problem, because it's hard to make molecules that are going to penetrate and do some damage to the bacteria. And one of the reasons why is because they've got these super strong cell walls. Peptidoglycan is super rigid, and it is a heteropolymer. It's got some two different flavors of monosaccharide. So remember that chart? There was a chart that showed all the different derivatives of glucose. We'll say glucose and all its cousins. And then there's a table that had all the different symbols that go along with the glucose cousins. So that's what's shown here. And one of the reasons why peptidoglycan is so strong is because in addition to these long polysaccharides, you've also got cross-linking by short peptides. And peptide bonds are also difficult to break. So it's a very strong heteropolymer. And that is one of the reasons why bacterial infections can be difficult to treat. Because if you're trying to, get rid of the bacteria, you have to have something that's going to penetrate or something that can chew up the peptidoglycans. And that's a whole different ball game. That's like a whole different class. But it's something for you to think about. This table summarizes some other polysaccharides that we did not go over, like dextran. You're not responsible for these, but just be familiar with them because these can be used to test your knowledge about, heteropolysaccharide versus homopolysaccharide in terms of the structure. I'm not going to ask you, what is chitin? Is it homopolysaccharide or heteropolysaccharide? Not going to do that to you without showing you some snippet of structure. But you know how we do case studies, or in the case of an assignment or an exam, you'll have kind of a new system to look at and interrogate using all the questions that you've learned to ask and all the vocabulary that we've built. So just know that those are fair game in terms of giving you something that you can answer questions about. So being familiar with, a little bit of how these things work already will give you a leg up. That's it. Thanks for watching. Make sure that if you're going to look at the book, again, you only focus on sections 1 and 2. Everything that I didn't cover in those 2 sections, you are not responsible for. This textbook is kind of above our pay grade in some respects. We're not going over every single detail because we simply don't have the time. So make sure that you only focus on what we covered in this video lecture. Be safe, and I'll see you in class.